Welcome to Click for IB Biology. We're studying genetics and we're looking at the process of meiosis at the higher level and considering the idea of unlinked genes and indeed alleles. There's a blank version of this PDF down in the description. You can download that, annotate that with your own notes, and then of course take the 10-question quiz, check out the answers to secure your own learning. So just a review of meiosis when we're looking at one homologous pair. So this is the situation where we have a genotype which is big A, little a, and of course we understand that that means that on one of the homologous pair we have the big A allele, and on the other one we have the little a allele. This is just, the genotype is just to for ease of identification. So we know that in the interphase we have big A, little a like this, and then following DNA replication there would be doubles of those in the chromatids like so. And that the key feature of meiosis 1 reduction division is the separation of the homologous pairs. So let's do that. We would have big A, little a, and so then we can see from my annotation that we have the separation of the alleles as well. So this is the key genetic feature of meiosis 1. Meiosis 2, on the other hand, when it occurs normally, simply separates the pairs of chromatids like so, and like so, and of course in the second one, resulting in 4 gametes where the diploid number has been now reduced to the haploid number from the diploid number of the parental cell. So we have the separation of the homologous pair, the separation of the alleles. So that's the part that we're trying to understand from the higher level perspective, and in the first step towards that understanding we're going to trace the we're going to trace the fate of two homologous pairs. So this one we have big A, little a, and this one here we have big B, little b. Now notice that that's one homologous pair and that's a separate homologous pair. So this is the meaning of the phrase unlinked. They are on separate homologous pairs. So here we have the chromosomes during the interphase, and of course we know that they go through DNA replication to look like this, and then we would expect to, in meiosis 1, we expect to separate the homologous pairs, and therefore to separate the alleles, and this is called the segregation of the alleles, and in fact was often thought of as Mendel's first law. So this will separate this way and this will separate that way. So you can see with my 2 cells here we have the blue chromatids and we have the green chromatids, and here we have the red chromatids and the orange chromatids. So that's a homologous pair and that's a homologous pair. However, it is equally possible that when we have the separation of the first homologous pair, red and blue, here and here, that the second homologous pair may separate in the opposite direction. And you can see that I've drawn that with the orange coming this way and the green ones going that way. And so this introduces a new concept. When we move from one homologous pair to 2 homologous pairs, we see that the separation of the second homologous pair is independent of the separation of the first homologous pair, and this increases the variety of potential gametes which will result from meiosis 1. This independent separation is known as the independent assortment of homologous pairs and is often referred to as Mendel's second law. Let's track this all the way through to the gametes. So now we're going to go through meiosis 2. which is the separation of the chromatids. And as we can see, this is no different to when we had one homologous pair, but with two homologous pairs, we simply end up with two from this one. That's that pair there, two from here, and two from there. However, notice that if we think of this as a ratio, we still have the ratio of 1 to 1 to 1 to 1. We have 4 different arrangements of the chromosomes in the gametes, and we have halved the chromosome number. This can actually be, this is true whether we, having learnt for 2 homologous pairs, the rule remains the same for 3 homologous pairs, and indeed however many homologous pairs that you have, is that we deploy the first law, the separation of the homologous pairs. That's the first rule. And the second rule is that homologous pairs, different homologous pairs, separate and segregate independently of each other, known as independent assortment. So therefore we can actually calculate just how much variety we will get with the gametes. And this is done by taking 2 to the power n, where n is the haploid number. Now in this particular case, the diploid number was 4. The haploid number therefore is 2. So 2 to the power 2, of course, is 4. And you can see above in the diagram, we have 4 different types of gamete. Now in human beings, if we take an example, our haploid number is 23. So we have 2 to the power 23, which I'm reliably told is 8,333,608 different possible arrangements of the chromosomes in a sperm cell or an egg cell. There are 8.3 million different possible arrangements purely on the basis of meiosis, independent assortment of the chromosomes. Now, of course, a quick calculation will show that if we take this one step further, then if we have the paternal side has 2 to the power 23 and the maternal egg production is 2 to the 23, then this means that we have a potential of 2 to the power 46 different possible fertilizations, which I understand to be somewhere in the region of 70 trillion different possible outcomes from 2 human beings having a child. There's approximately 70 trillion different arrangements of their chromosomes purely on the basis of the rules of meiosis as we've seen them so far. Let's apply this to a dihybrid cross. The methodology for working out the different gametes can be simplified a little bit, and so let's do that. So what we have to do is we have to separate the homologous pairs, and then so that's the first Mendelian law, and then having done so, we then separate big A, big B, little b independently of each other. So let's take a look at that. So we can have big A can end up with the big B, so our gamete could be this, A, B, or we could have big A with little b, big A, little b. This is purely from meiosis. Then we can take little a with big B, so little a, big B, and little a. I'm sorry, drawing's getting complicated now. A with little b, like so. And so here we have from this dihybrid genotype, we are creating through meiosis 1 and meiosis 2, we are creating 4 different gametes. We're going to go on in later parts of this course to look at the consequences when we introduce a second individual and look at the possible fertilizations which can arise from these crosses. Links to those particular crosses are now appearing here in our next video. There's a chance to subscribe to the site, and of course, the playlist for genetics videos.